Hello, everyone, and welcome to the Simons Searchlight CHAMP1 Family Conference. We have 3 presentations for you today that will all be done in a row. After the 3 presentations, we'll have plenty of time for a Q&A with Dr. Wendy Chung. Even though the Q&A is at the end, you're able to submit questions at any time during the presentation using the Q&A box at the bottom of your screen. Also, make sure to stay tuned. At the end of the conference, we'll be opening up the video and audio for attendees so that families can connect with each other. Our first presentation today is a recording for Genetics 101 and How to Read Your Lab Report from Rebecca Smith and Julianne Savat. Rebecca is a genetic counselor for Simons Searchlight, where she works to review participants' lab reports and to answer family questions regarding everything genetics. Rebecca also works at the Autism and Developmental Medicine Institute, helping to make meaning out of genetic testing results with families. Julianne Savat received her Master of Science in Genetic Counseling from University of North Carolina, Greensboro in 2015, and started her career as a clinical genetic counselor and research coordinator at Geisinger. During her time at Geisinger, Julianne has worked with families in the Autism Developmental Medicine Institute and cancer genetics clinics. Julianne is currently a member of the Clinical Genome Resource Team, where she coordinates Genome Connect, a patient registry for individuals that have had genetic testing, and is involved in research efforts using Geisinger's MyCode Biobank. Julianne is motivated to help increase our understanding of genomics and to empower patients to be active partners in genomic discovery. So here is their presentation.
Hello, thank you for joining us today to talk about Genetics 101 and how to read your genetic testing lab report. My name is Julianne Savat, and I will be providing you with an overview of some of the common terms you might find on your laboratory report, and then my colleague, Rebecca Smith, will be actually walking through an example of a laboratory report to help you sort of put all these pieces together. So today I want to begin with an overview of genetics, and I know that for some of you joining us, these concepts and terms might be familiar to you. For others, these might be new terms to you, or this might be a bit of a refresher from whenever you met with your physician or genetic counselor. My hope is to review chromosomes, DNA, and genes to discuss types of genetic changes or variants, types of genetic testing that you might have gone through, and then some other common terms that you might wish to know or that you might see on your laboratory report. And then, as I mentioned, Rebecca will walk through how to review your laboratory report using an example to walk us through all of these terms. One topic that we won't be touching on today, but that we get a lot of questions about, is gene therapy. Dr. Wendy Chung's plenary presentation on August 14th at 2 p.m. will provide much more information about gene therapy, so be sure to attend that presentation if you have questions around this topic. But now on to an overview of genetics and some of the terms you might see on your testing reports. All of you listening have gone through the genetic testing process, and you might have spoken to a genetic counselor or geneticist, so as I said, some of this might be familiar, but my hope is to break it down and to help give you a bit more information as you re-review or walk through your testing report. Each one of us has more than 1 trillion cells that make up our bodies. We have different types of cells, including our skin cells, our brain cells, our heart cells, and these really are like the building blocks of our bodies, sort of like how bricks make up a house. In each and every one of those cells, if we crack it open and look inside, is our genetic material, or our DNA. And our DNA is like our body's instruction manual. Each instruction is called a gene, and in fact we have so many instructions, so many genes, that I often say that our genetic material is like a shelf of instruction manuals. To keep our DNA organized in our cells, it's wrapped up very tightly into these structures called chromosomes. Typically, people have 46 chromosomes that come in 23 pairs. Our chromosomes come in pairs because we get 1 in each pair from our mother and 1 in each pair from our father. The first 22 pairs of chromosomes are the same in males and females, and they're really organized based on size and named based on size, with the first pair being the largest down to the 22nd pair, which is the smallest. The 23rd pair of chromosomes, that last pair, are called our sex chromosomes. Males are typically XY and females are typically XX. As I mentioned, each of these chromosomes is made up of DNA that's wound up very, very tightly, almost like a ball of yarn. Our DNA is what provides the actual instructions for our body, and it's actually instructions for how our body can make proteins. Each instruction is a gene, and each gene codes for a specific protein. We each have approximately 20,000 genes that we inherit from our mother and father, and these help our bodies grow, develop, and work. Each protein has a specific role, or maybe a couple roles within the body. Some determine our eye color, others help our heart grow, and others are important for how our brain develops. Our body spells out the instructions in our genes using the letters A, T, G, and C. And our body actually reads through those letters to use our genes as instruction to make those proteins. When reading the letters within our genes, our body actually reads those letters in sets of 3. We call that the triplet code. A set of 3 letters in our DNA codes for one amino acid in a protein, and amino acids are just the building blocks of a protein. So a protein is made up of multiple amino acids, and our body would read 3 letters at a time to make one amino acid. So in this example, our body would read ATG to make this first amino acid, the letter CAC to make the second amino acid, and so on. There are 20 different amino acids that make up our proteins, all coded by these 3-letter sets. So now that we've talked about DNA, chromosomes, genes, and proteins, let's talk a little bit more about genetic variants and what they are. Genetic variants are changes in our DNA. While most of our DNA sequence is the same from person to person, so myself and Rebecca, most of our DNA is the same, we all have genetic changes that make us who we are. Some variants just help make us who we are, make us individuals. Others affect how proteins work in the body and might cause developmental or health differences. There are 2 main categories of genetic variants that we want to focus in on and talk a little bit more about today: copy number variants and sequence variants. Starting with copy number variants, these are genetic changes where a person has a large piece of missing or extra DNA. It's often thousands of extra or missing letters within their DNA. If a person is missing genetic material, that's called a deletion. If they have an extra piece of genetic material, that's called a duplication. So if we think about our genetic material, our DNA being a shelf of instruction manuals, it's almost like a copy number variant is almost like a person is missing books or has extra books within their DNA, their genetic material. Copy number variants are typically named for their location or where they are along those chromosomes. So for an example, we might see a report that says 6p24.1 deletion. This helps us know where in a person's genetic material this missing piece of DNA is. The 6 tells us that this missing piece of genetic material is on one of this person's number 6 chromosomes. When we talk about chromosomes, we divide them into 2 parts, the P arm and the Q arm. The P arm is the shorter arm, the Q arm is the longer arm, and they're divided by this little pinch point here, or what we call the centromere. So 6P just tells me we're on the smaller arm on the chromosome 6. And then the 24.1 tells us exactly where we are along the chromosome. You can see that the chromosomes have a banding or striped pattern, and this is actually what they look like under the microscope. So the 24.1 tells us exactly what band or stripe has the deletion. So again, copy number variants help tell us where that missing or extra piece of genetic material is. Deletion tells us that a piece is missing. Duplication would tell us that we have an extra piece. The other type of genetic variants I want to focus in on today are sequence variants. Sequence variants are genetic changes where there's a difference to the letters within a gene. Sometimes this is a single letter or maybe a couple of different letters. And if we think about our genetic material as that shelf of instruction manuals again, a sequence variant is like there being a spelling change in one of the sentences in one of the books. So a small change, just one or a couple of letters that's different. Sequence variants are typically named based on the change to the DNA and the change to the protein. If you see a C period or a C dot on your report, this tells you what the change to the DNA is. That C period is often followed by a number, so this tells us exactly what letter is changed in our DNA. So in this example, we're talking about the CSNK2A1 gene, and we're talking about letter 593 within that gene. So if we start at 1 and count up to 593, that's where we're talking about. The first letter, A, in this example, is what letter people typically have at position 593. The greater than symbol is our way of saying is changed to, and then that second letter, G, tells us the letter in this individual. So altogether, this tells us that at letter 593 in the CSNK2A1 gene, most people have an A, but in this person, they have a G. This genetic change can also be described by the change to the protein. So if you see a P period or a P dot on your report, this is describing what happens to the protein or what change there is in the protein. This P-lyse 198 arginine tells us that an amino acid 198 in that protein, so if we start at 1 and count up to 198, that's the place where we're looking. There's typically a lysine, and this is the abbreviation for lysine, one of the 20 amino acids. But in this individual, they have an arginine instead. So if you're reviewing your report, if you see that C period or that P period, this tells you whether you're talking about the change to the DNA or the change to the protein, and you can break down those pieces after that C period or P period to know a little bit more about what the genetic change is. There are several types of sequence variants that I want to mention and talk through since you might see these terms on your laboratory reports. The first is missense. Missense variant means that one amino acid, one of those protein building blocks, is substituted for another based on a single letter change to the DNA. So in that example that we just walked through, the amino acid at position 198 in the protein is typically a lysine, has been changed to an arginine. One amino acid was changed. So this is an example of a missense variant. Another type of sequence variant you might see is a nonsense variant. Like missense variants, nonsense variants are caused by a change in a single letter in the DNA. Here we can see that at letter 265 within the SETBP1 gene, most people have a C, and there's a little error here. Forgive me, this person has an A instead. In addition to coding for amino acids, the 3-letter sets in the DNA also help our body know when to stop a protein. So sort of like a stop sign that says this protein's done, stop making it. A nonsense variant happens whenever the DNA change results in an early stop sign or an early stop signal. So the protein is shorter than typical, and this can impact how the protein works in the body. If you're looking at your report, you can tell if it's a nonsense variant in that P period or that description of the protein change. There'll be either a TER, short for termination, a star, or an X to note that there's an early stop signal. You also might see sequence variants that result in a small number of extra or missing letters within the gene. Insertions or duplications add a letter or several letters to the DNA sequence, like this example here, where there is at position 6073, we typically have an A, and this person has a duplication or an extra A inserted there. A deletion removes a letter or several letters from the DNA sequence. Now, sequence variants that have insertions, duplications, deletions are different from those copy number variants that we talked about a few slides ago, because these only involve one or a small number of letters within the DNA, while those copy number variants that we talked about often involve thousands of letters within the DNA. So to help sort of make that distinction there, it's just how many letters are involved. Adding or removing letters in the DNA sequence can result in what we call a frame shift variant to the protein. As we talked about earlier, the body reads that DNA sequence in sets of 3 letters. If we duplicate just one letter, like in this example, it will shift how our body reads the remaining letters and can change the remaining amino acids in the protein and eventually lead to an early stop codon. You can identify frame shift variants on your report because they typically have the letters FS in the description of the protein change. Those insertions, duplications, and deletions of one or a few letters can cause these frame shift variants. So now that we've talked about genetic variants, so copy number variants and sequence variants and some of the different types of sequence variants, what sort of genetic testing might be used to identify these changes? All of you listening in today likely are familiar with one or more of these tests from your own genetic testing journey. And all of these tests are typically done using blood or saliva samples, as you might know. The first test that I want to talk about is karyotype. A karyotype is a test that we used to use quite frequently, but we use a little less often now. And it's a test that actually looks at the chromosomes under the microscope. It can identify large copy number variants, so large missing or extra pieces along those chromosomes, or even a whole missing or a whole extra chromosome, like an extra chromosome 21 that causes Down syndrome. So this is sort of the most zoomed out view of a person's genetic material that we can take. If we zoom in a little bit further, we can do a test called a chromosomal microarray. And this test can detect smaller copy number variants, so smaller missing or extra pieces compared to the karyotype. If we want to actually read through the letters of someone's DNA, we have to zoom in even more and use a technology called sequencing. Gene panels use sequencing to look at variants in a specific list of genes that are tied to certain health conditions or developmental differences like seizures, autism, developmental delay. Another type of test that uses sequencing is called exome sequencing. Exome sequencing uses sequencing to look for variants in all parts of our genes that provide our body with instructions. These parts of genes that provide us with instructions are called our exons, and if we look at all of them, we call that our exome. So exome sequencing looks at all of those pieces of the body that provide us with instructions for our proteins. So whenever you have genetic testing, if you identify a genetic change in a specific gene or a copy number variant that involves maybe one or a few genes, the next step for the genetic testing laboratory is to interpret that variant or to try to determine if it impacts how the gene works in the body and if it could be associated with a health condition, a developmental difference, or a risk for a condition in the future. As I mentioned earlier in my talk, we all have lots of genetic variants. Many of them just make us who we are, and only some will impact our health and development while others don't. So when a lab sees a genetic change, they try to use all of the information that they have available to them to try and determine if that genetic change might be associated with health or developmental differences. This means that they look at things like, how does that genetic change impact the protein? Does it cause a frame shift? Is it a missense variant? They look at where it's located in the protein. Is that a piece that's important for that protein's job in the body? They look to see if that genetic change has been seen in other children with similar symptoms or health concerns. They look to see if it's been seen in the general population. And then they try to put all of this information together to assign an interpretation to that variant or what they think about its relationship to health. Interpretations from genetic testing laboratories primarily fall into three categories: benign, uncertain, and pathogenic. Benign means that the copy number variant or sequence variant is not known to cause any significant health or developmental differences. Pathogenic means that this copy number or sequence variant is believed to cause symptoms or features. If there's not enough information to put a variant into those categories where we say, yes, it's tied to health or development, nope, we don't think it's related, we have to call it uncertain. If we don't know enough to say one way or another, we say that's a variant of uncertain significance, or a VUS. This means, again, that there's not enough information for us to say if the variant is tied to health or development, and that more information is needed. Our hope is that over time, we'll be able to better understand that uncertain result and to put it into a more concrete category, but sometimes that just takes time. We need to learn more. Other times we can recommend or offer things like parental testing to help inform uncertain results. If parental testing or testing mom and dad wasn't done as part of initial testing, this can help clarify things in some cases. Understanding if the variant was inherited from a parent or if it's new and unique to the child can help us better understand that variant and its link to health or developmental differences. So on to some other common terms you might see on reports. So first, on your reports, you might see the terms autosomal dominant, autosomal recessive, and X-linked. To walk through these terms, I wanted to first return to this slide that you saw a little bit earlier today. As a reminder, we all have 2 copies of the first 22 pairs of chromosomes. We have 2 copies of each because we get one from our mother and one from our father. So for all of the genes that are packaged into these 22 chromosomes, we typically have 2 copies of those genes, one from mom, one from dad. Again, our last set of chromosomes are our sex chromosomes, with females typically having 2 X chromosomes. So they typically have 2 copies of all of the genes that are packaged into those X chromosomes. And males typically have an X and a Y, so they have 1 copy of the genes that are on that X chromosome. Autosomal dominant and autosomal recessive are used to describe conditions caused by changes in genes that are on those first 22 pairs of chromosomes. Conditions that are caused by having a pathogenic variant in 1 copy of the gene are called autosomal dominant. If you need to have a pathogenic variant in both copies of the gene, this is called autosomal recessive. Conditions caused by pathogenic variants in a gene on the X chromosome are called X-linked. Symptoms can differ between males and females because females typically have 2 copies of this gene, while males typically just have 1 copy. They only have that 1 X. Sometimes autosomal recessive, autosomal dominant, and X-linked are called inheritance patterns. But these alone don't tell us if a genetic variant was inherited from a parent or whether the variant was found on 1 or both copies of the gene. They're really just used to help us understand if having 1 pathogenic variant causes a condition, or if you need to have pathogenic variants on both copies of a gene to cause a condition, and whether the gene is located on those first 22 pairs of chromosomes or on that last pair, those sex chromosomes. The words heterozygous, homozygous, and hemizygous that are seen on some genetic test reports tell us about the specific individual who was tested. If the genetic variant was found on 1 copy of a gene, we use the term heterozygous. Homozygous tells us that a person has the change on both copies of their gene, and hemizygous is used to describe a male that has the genetic variant on his only X chromosome. On your report, you might also see the words inherited or de novo, and these terms help us to understand if a genetic variant was inherited from a parent. We determine if a genetic change was inherited or new to a child by testing the parents for that variant. If the variant was passed on, so we see it in mom or in dad, we say that that genetic change was inherited. As many of you might know, some genetic changes are new and unique to a child, meaning that they weren't inherited from mom or dad, but they happened early on in the sperm, in the egg, or early on in development. Whenever a change is new and unique to a child, we call these de novo variants, because de novo means anew in Latin. So it's an old Latin term that's sort of stuck in genetics, but it just means new and unique to the child. Finally, a few families might have the words mosaicism or mosaic noted on their report. Mosaicism means that only some of a person's cells have the genetic variant. Some cells have the variant, some do not. For some families, this might have been identified through their testing, that when looking at a blood sample or a saliva sample, the genetic testing laboratory only saw the genetic change in some of the cells. With our current testing technology, if we detect mosaicism, we often can't say which cells in the body have the genetic change, or really how many cells in the body have the genetic variant. But this is something that might be reported on some people's reports, so I wanted to be sure to mention it. Sometimes we also might come to learn that a parent has a specific type of mosaicism called germline mosaicism. This means that more than one of a mother's egg cells or more than one of a father's sperm cells, what we call their germline, have the genetic variant. Our current technology doesn't allow us to detect germline mosaicism through our testing of blood or saliva samples, but sometimes this is identified when parents have more than one child with the same genetic condition, but they themselves don't have the variant found in their blood or saliva samples. So we might think that the genetic change is new or unique to the child, but we come to learn that a parent has the genetic change in multiple of their sperm or eggs after they have multiple children with the same genetic variant. If a variant is found to be de novo in a child, so we find it in a child, test mom and dad, and neither of them have it in their blood or saliva sample, we say that there's less than a 1% chance for them to have another child with that same genetic change. Again, a family might have more than one child with that genetic change because of this germline mosaicism. So now that we've walked through many genetic concepts in a relatively short amount of time, I want to turn it over to Rebecca to walk through an example report to help put some of what we talked about into a bit more context, to walk through a report with you, to try and drive some of these terms home and help you walk through your laboratory reports. And then of course, we'll be available for questions after we walk through an example.
Great, thank you for that wonderful background, Julianne. So like she said, one of the most common questions that we get here at Searchlight is, how do we read our genetic testing report? And at first look, it can certainly be overwhelming to try to make sense of all these parts and pieces, but we do hope to break it down for you in a meaningful way where all of these parts can be understood. Lab reports from different labs may look slightly different from each other, but they're all made up of the same parts. We'll use an example today from one of the labs we see reports from most often, called GeneDx. And so what's a better place to start than at the top? At the top, you'll see all of the demographic information. So your child's name, birthday, the type of sample that was used. So that may be blood or a buccal sample, which would be like a cheek swab. You'll also see the dates of the whole genetic testing process. So when that sample was collected, when it arrived at the lab, and then when the report was made. And this is important because we're learning more and more about genetics every single day. So if this report is over 2 years old, well then maybe it's time to ask your genetics provider to review it, because we may know more information now than when the report was initially made. And then moving down, the next thing that you'll see is the test that was ordered. And we already covered this, but I'd like to review it just again. There are 3 main types of testing that we see here at Simons Searchlight. The first result that we can see are on chromosomal microarrays. So this is that test that's looking for any missing or extra pieces that we call deletions or duplications. We know that this change in the amount of somebody's DNA can change how their body works and can result in things like medical and developmental changes. You can see here the list of copy number variants that this test would find that we accept here at Simons Searchlight. Moving on, other types of testing we can see are both gene panels and whole exome sequencing. And as a reminder, these are the tests that read through the letters of the DNA. Gene panels are looking at specific genes that we know are related to a specific health or developmental condition. So some people may have had panels looking at genes related to epilepsy or autism and developmental differences. Other individuals may have had whole exome sequencing, and that's reading through all of the letters of all of their genes throughout their DNA. And this type of testing often would result in those sequence changes that we can see in any of the genes listed here. And so now, moving farther down, the next section is often called clinical indication. It may also be called relevant history or pertinent information. And this really just describes why the child is having genetic testing. It's often just a brief summary of their medical and developmental history. And then we move down and we get to the results of the testing. And this is often the meatiest section. So we'll break this down piece by piece to review what all of these different parts mean. So we'll start on the left, and this is where we'll find the gene name. As we know, many genes in our body help our body work like it's supposed to. And this is an important first step in thinking about what any of these changes may do. We think, well, what does this gene do in our body? What instructions does it code for? Many of the genes that we see in Simons Searchlight, for example, may help with brain development and brain growth. And so if we have a spelling change that breaks how this gene is working or changes how it works in our body, it makes sense then that it could lead to things like developmental differences. And then we'll move to the next box, which is labeled disorder or disease. And this tells us the name of the condition that results when somebody has a spelling change in that gene. This name is most helpful if you'd like to learn more about what it means. This is the name that you would type into a search engine, for example, to look up more information. Down in the sections below this box is often a summary and interpretation section. That's when you'll find the most details about what this specific condition actually is and what are those associated signs and symptoms. All right, and continuing on, the next box is labeled mode of inheritance. And for this section, we're jumping back to information we already reviewed today, but I'd like to cover again. So as a reminder, we all have 2 copies of each of our genes. We inherit one from our mothers and one from our fathers. It's totally random what copy we get from each of our parents and ultimately what copy we may pass on to any children. And our body uses both of these copies to work like it's supposed to. There are 3 main types of conditions that we see at Simons Searchlight. The first is called autosomal dominant, and this is most of the changes that we see here. This means that of somebody's 2 copies, 1 has a spelling change, and that's enough to affect how their body is working. We can also see autosomal recessive inheritance, and so this is when both copies of that spelling change are present on both of those genes. Oftentimes, with these autosomal recessive conditions, each parent only has 1 gene change. They're called carriers, and most times, those mothers and fathers don't show us any signs of the condition. It's only when both changes are passed down to the child that we see signs of the genetic changes. And then finally, we have changes that can be inherited on our sex chromosomes, so those X chromosomes. We call that X-linked inheritance, and as we reviewed, males typically show more significant signs and symptoms if they have a gene change on that X chromosome because they don't have that backup X chromosome that females do. All right, so we're going back to our report and making our way through. We'll move on to our next box, which is labeled variant. And this tells us that very specific gene change that we found, that spelling difference that we can think about both in terms of the DNA letter that's changed, but also the difference on the protein level and how that amino acid is changing. And then we'll continue on to zygosity. And this is going back again to that autosomal dominant and recessive. This is telling us how many changes somebody has. So again, if somebody is heterozygous, that means that there's a change on one of their chromosomes. Most often, this relates to autosomal dominant conditions. If somebody is called homozygous, that means they have those two gene changes, and that most often corresponds to autosomal recessive conditions. And then when we get to the box that says inherited from, that's really getting at the question of, well, where did this gene change come from? Sometimes these gene changes are inherited from a parent. And depending on what the specific change or variant is, that parent may show us the same signs as their child. They may show us different signs or symptoms that their child doesn't, or they may show us no signs at all. This is also how your provider would be able to tell you, well, what are the chances that any other or future children would have this same genetic change? Many families may be interested in this information for family planning. It's also possible that these gene changes may be brand new, only in the child, and was not inherited from a parent. This is what we call de novo. And in these cases, the chance for any other or future children to have the same change is often quite low. And it's very important to remember with these genetics that these changes are totally random by chance changes that occur. There's nothing that anybody does or doesn't do that causes them. And we also have no control over what DNA we pass on to our children. And then lastly, we get to classification. And this is how your medical providers determine what the change is or isn't doing in somebody's body. And like Julianne reviewed for us, there are 5 main classifications. And these labels just really have to do with how confident are we that this change is the answer for that child's developmental or medical differences. Most often, changes that are benign or likely benign aren't reported because we think that's not our genetic answer. We can also have changes that are called VUSs, or variants of uncertain significance. These are essentially inconclusive results. And these are those genetic changes that we just don't have enough research yet to know exactly what they mean. But we hope that in the future, as we learn more, we'll be able to provide these families additional information as to whether or not this change is that child's answer. And then finally, we have pathogenic and likely pathogenic gene changes. And these are those results that we're confident are changing the way that that child's body is working and is our genetic explanation for why those child has those medical or developmental differences. And then as you look to the bottom of your report, you'll find these usually large sections titled Interpretation and Summary. And this is really where you get a lot of the information about, well, what is this gene doing in the body? And when there are gene changes, what have we seen? What are associated medical findings or developmental differences that we see in other individuals? Your provider will also be able to use this information to guide management moving forward, whether that means seeing other providers or keeping your child enrolled in all of the services and therapies that they're already involved in. All right. And now it is possible that some of our families here today may have had a chromosomal microarray report. And so I wanted to just briefly mention it right here to show you that this is what it looks like. While many of the pieces are similar, it does have some different sections. But I just wanted to bring it up to say that if you have one of these reports, we are more than happy to walk it through with you because it's possible that somebody could have a deletion or a missing section that encompasses one of the genes we're talking about today. So don't hesitate to get in touch with us if you'd like to walk through these parts and pieces as well. All right, and we've made it to the end. So we hope that today's presentation has been helpful in making a little bit more sense and meaning out of these really complex genetics. If you have any additional questions specific to your own child's report, please don't hesitate to reach out to us at our email here, and one of our genetic counselors will be in touch shortly. Thank you for watching.
All right, so that was our first presentation. Our next presentation is going to be by Dr. Wendy Chung. In this recording, Dr. Chung gives you an overview of the questions you may have regarding gene therapies. And just remember to submit the questions that you have in the Q&A box found at the bottom of your screen. We won't be able to answer the questions right away, but Dr. Chung will be on later to answer all of the questions. And if you don't want to forget the questions that you have, you can just put them in now. And with that, we will move on to Dr. Chung's next presentation.
Hi, I'm Wendy Chung. I'm going to tell you this morning about some exciting advances we have in gene therapy. So your child or your family member's condition is genetic, and the obvious question becomes, can we do something to fix that? Can we go in and edit the gene? Can we fix the gene? Can we add back a gene? And many of you have been asking that question. I'm going to give you an update today in terms of where we are. I certainly am optimistic that in the future there are going to be many options in terms of genetically and molecularly based therapy, but really the million-dollar question is when. And I don't have a great answer to that, but I will say that there are many people working on this, and I'm going to try and demystify this a bit for you today by explaining some of the strategies. So on the left here is what we're showing as a cartoon of a normal gene. That gene encodes the instructions to make a protein. And again, on the left here is someone where that gene is working fine. Now on the right, there's a gene that has a genetic change. It's shown as a broken gene here. And in some cases, it makes no protein. That gene just doesn't have instructions that work, so it makes no protein. In other cases, it may make a protein, but it looks different than the normal protein. And in some cases, this can lead to an individual with a genetic condition, and that's what we're going to be talking about today. So very broadly, depending on whether or not the problem with the gene is not making a protein or making an aberrant protein or a protein that looks different, there may be slightly different strategies. I'm going to go through the most common strategies that I think might work for your family member's gene, but to say that there are still evolving strategies. And so I won't have time to go over in all of the strategies or in great detail, but I'll go over the most common ones. So let's dig down for a second. So here on the left, shown in red, are individuals that have a gene that has this particular change in it, a change that may, in this particular case, result in disease, and it's because this protein is not made. In those cases, there can be different strategies, but strategies in which we add back a normal copy or a functional copy of the gene. This then makes protein that was missing or potentially not there in enough quantity, and then corrects the condition because we have added back what was missing. Now there's some subtleties in this. As we add back this functional copy of the gene, where does it go? Does it go directly within the genome or the genetic information of the person? Does it integrate or does it become part of them? Or is it something that's living off to the side in some way? And there are some important differences between that that have to do with whether or not we get it in the right place and whether or not it stays around for a long period of time. So those are some subtleties, but this is the basic idea. Now, in certain cases, I showed you in that picture, there may be a gene with a change depicted here. And in this case, it actually does make the protein, but in this case, the protein is not doing what we need it to do and may in fact disrupt or interfere or gum up the works of the regular gene. So it's doing something that we don't want it to be there doing. And in that case, we actually have to get rid of it. It's not simply replacing it or adding back, but it is actively getting rid of this red protein here. And so there are strategies. I'm going to use the term gene silencing, but it's one way or another of being able to knock down or get rid, and if we can, of doing it specifically for the form of the gene that's making the protein that we don't want to have there. And so in that case, the reason why we get correction is we are getting rid of this particular protein. Now, for doing that, there are different ways of being able to deliver what look like these little, I don't know, these little green ladders here, or half of a ladder. There's a way of being able to deliver it to the cells in various ways. I'm not going to focus on those details today, but it is critically important to know the difference between the case I described over here, where the gene, or the protein rather, is missing, and the case over here where the protein is present and we don't want that version of the protein there. Fundamentally very important to differentiate. And I'm glad to say that in the cases that we're going to be talking about for your gene, I think we're to the point of understanding those differences. So that's the good thing. Now, some of you have probably jumped to the obvious conclusion to say, well, why don't, if the gene with the changes here, just fix it? Like surgeons, just go in and snip it out, take out the version that's not working, sew in the version that does work, edit it, correct it, make it whole again. And if we fundamentally correct the DNA itself, then it will correct the protein. We don't have to do anything fancier than that. We can just go in and get to the root cause or get to the heart of the problem. And so this is a technology people are using called gene editing. You may have heard the terms CRISPR, Cas9. Those are effectively like the molecular scissors to be able to go in and to find just the right place, just the right location to do the snipping and then to do the sewing in. This is certainly a technology that is evolving very rapidly. Some of you have heard about the improvement recently called prime editing, which increases the fidelity or the likelihood, that the editing gets done in just the right place. Now, the opposite of that would be some editing that's promiscuous, where you don't get to just the right place, but you may get to someplace down the road that you didn't mean to be editing, or someplace down the road over here that you didn't mean to be editing. And there is the possibility of misediting, actually putting in something or cutting and pasting in the wrong place, and that could actually be bad. That could actually disrupt a different gene that you weren't intending to touch and cause its own independent problems if it's not very precise, very accurate. So within this, as I said, there are more and more tools that are becoming available. Some of these use what we call vectors, or ways of being able to deliver these tools to cells. I'm going to be talking about that in just a second. Some of them are delivering these tools, these scissors, the thread to be able to sew this in. And really, the good news is that as these tools are developed for one condition, they can be readily applied oftentimes to other conditions. So you may be hearing about methods that have to do with gene therapy in the eye, or gene therapy for sickle cell disease, or gene therapy for a liver disease, or a bleeding condition. The good thing is that we are going to shamelessly beg, borrow, we won't steal, but we will borrow from others in terms of, as they develop those tools, we will use those same tools that we're thinking about for your family's condition as well. So we do have to be patient. And what I want to give you is a little bit of historical perspective. You don't need to read all of the details here, but simply to show the timeline. So when we first started thinking really in the modern era about gene therapy was about the time I started my career in genetics, about 30 years ago in 1989. Now, the good news is we've come a long way in the last 30 years. We've made major advances in being able to understand this, but we also have learned some very valuable lessons. And I'll just point out one valuable lesson we learned in 1999 was that this is something that could actually do harm, that we need to be careful about this because a young man by the name of Jesse Gelsinger was actually, unfortunately, passed away during his gene therapy for a different condition. From that, we became, as a community, internationally cautious in terms of being able to be careful, be safe, have checks and balances, test things in cells, test things in other animals, but really to make sure that we were going to do no harm, that we were going to protect the safety of the individuals who went through gene therapy studies. And because of that, we've slowed down. We're not running quite as fast, but on the other hand, I can assure you that we're always have the participants' best interest in heart as we're taking those precautions. So let me tell you a little bit about how we get those genes into the cells. And there are some limitations based on these delivery mechanisms or these delivery systems that we have. So the first is that there is a virus called adeno-associated virus. And I'm showing you here this viral particle. Within this is something that is a virus that is part of what causes the common cold. We have done things to these, genetically engineered them so that you can't pass them from one person to another. So it's not like the cold, it's not like SARS-CoV-2. This is not something that gets infected from one person to another. We have basically paralyzed them so that can't happen. But they are good delivery systems that inside this little squiggly here is the gene that we're trying to insert. And so as we're doing this, as I said, we have inactivated these viruses. Importantly, this is a cell that I'm showing here, and this is the nucleus inside the shell that houses the genetic information. And these are meant to be chromosomes housing the genetic information. When this virus goes in and infects the cell, this genetic information for the new gene comes into the nucleus, but importantly, it does not integrate within the person's genetic information. So we call these non-integrating. By setting them up so that they are not integrating, they will not disrupt other genes that the person has. And one of the types of genes that we worry about in terms of disrupting are genes that are mission critical to the way your brain works or your heart works, and also genes that might protect you from cancer. We don't want this to result in changing one condition or trading one condition for another. We also have engineered these cells or used particular adeno-associated viruses that they hone in to particular parts of the body. And in fact, it's been quite successful, remarkably successful, at being able to hone into brain cells or to neurons. And that's been a huge advance. This is also something that works quite well for cells like brain cells, like neurons that are no longer dividing. Otherwise, if they were dividing, you can imagine this DNA that gets in would be diluted out over time. That is that when this cell divided, you'd still only have one copy of that DNA, and therefore only half of the cells that would retain that DNA and make the protein. So because brain cells don't divide, this is a very good strategy. It stays there. As we have gone through these gene therapy trials, and this is true across many different conditions, part of the reason why it takes so long is because we are, as I said, trying to make sure that safety is paramount. It's a very, very important thing to us. And as we do this, we try and make sure that we get just the right dose of the virus. We don't want to give too much to the virus. We learned our lesson. We gave too much. Collectively, we as a community gave Jesse Gelsinger, the young man who died, he got too much of that virus. So we're very careful about that. We're careful about testing, as I said, in cells and what we call preclinical testing in animal models. We test to make sure that the gene gets there at high enough dosages of the genes, and then ultimately not just that it's safe, but that it actually works. It's efficacious. It has the desired beneficial effect to slow down the disease or even to be able to reverse the disease. So a lot of thought goes into this to doing it well. Now the success, the good news is, and I'm just going to cut down to this bottom two lines, there are already 2 medications approved, 2 gene therapy medications approved for 2 different conditions, one an eye disease or a retinal disease, another for a condition called spinal muscular atrophy or SMA. And I'm very happy to say I've personally been part of these SMA studies, and we can now treat babies, newborn babies in some cases, with the gene therapy. It's literally an intravenous infusion through a vein. We don't even have to do a direct injection into the brain. We can just give it into the vein as a one-time treatment. I hope that this is going to produce a durable effect, that this is going to be something that's going to last for decades and hopefully a lifetime. We're still only a few years into this, but it does already show to be quite promising. Although I'm not going to go into it today, there are many, many other conditions that are currently being studied in what we call clinical trials. In other words, we're still testing them. Other brain conditions, so importantly here, adrenoleukodystrophy, another brain condition that I think is going to give us significant experience in terms of how we can do this for neurological diseases. So coming to wrapping all of this up, there are many things that we consider, and we try and matchmake. We try and find for any one disease, understanding the mechanism of how does that protein have its effect based on the way that that protein is working or not working. Think about exactly what type of technology would be useful to be able to get the correct version of the gene in, be able to permanently keep it within the brain cells, and to make sure that it's safe. Now, as we're doing this, there are a few other additional things that we consider. We want to be able to get in just the right amount. So it's kind of like Goldilocks and the Three Bears. We don't want it to be too much. We don't want to be too little. We want to be just right. And that just right, we hope, is what's going to be both safe and efficacious. And that's still part of what we're working on as we're getting into brain cells, making sure that we're getting just the right amount, making sure that we're getting in the brain, we're uniformly getting it into all the cells across the brain. There may be some consequences if it's a patchwork, if it doesn't look like we get into all the brain cells uniformly, we're not sure what that impact is going to be. And so we do need to be thoughtful in terms of how to do this, as I said, evenly, make sure it lasts, make sure there are no adverse consequences or no complications. So as we're doing this, just to let you know, there have been folks that have been at this for decades. The good news is that I can feel a real acceleration in terms of the breakthroughs that we're having, being able to learn from other diseases, other neurological conditions, and importantly, the safety that we now see in terms of the types of vectors we're using, the viruses we're using to deliver this, and importantly, that this is something that looks like it is lasting so far. I'm getting, for all of us, excited that we need to get ready. We need to prepare so that when some of these breakthrough technologies are able to scale and be applied to other conditions, I want our communities to be front and center. I want us to be at the starting line, ready to go, ready to take off on this race. And so many of the things you're hearing about at this family meeting are how we as a community can prepare for that day. I don't know whether that day is going to be in 2021 or 2025. It's hard for me to say, but I do think it's going to be here much sooner than we might think because we have made so much progress over the past 3 decades. So I'll stop here, but enjoy looking forward to the discussion.
Okay, and then our last person today will also be from Dr. Wendy Chung, providing the Simons Searchlight CHAMP1 registry update. Dr. Chung is an ABMG board certified clinical and molecular geneticist with over 20 years of experience in human genetic research. She's the Kennedy Family Professor of Pediatrics and Medicine and the Chief of Clinical Genetics at Columbia University, the Director of the Clinical Research at Safari and the Simons Foundation, and the Principal Investigator for Simons Searchlight. Welcome, Dr. Chung.
Thanks, Lindsay. Can you just tell me, are you seeing my slides okay? Or are you seeing them in presenter mode?
We're seeing the presenter mode if you wouldn't mind swapping them.
Okay, is that okay now? Okay, perfect. Okay, thank you for having me. So I know there is a question in the chat. I think what I'll do is I'll go through my presentation on the medical aspects, and then I'm glad to answer the question about gene therapy and any other questions that come up. So I wanted to use today to give the community an update. I'm sorry we couldn't be in person, but I do think it's important for us to continue letting you know what's been going on from all the work that you've been doing. So many of you know from previous meetings that we've continued to, although we've changed names from Simons VIP to Simons Searchlight, We continue to be very committed to the community of rare neurogenetic conditions that are sometimes associated with autism, and to be able to provide information to the researchers so that you don't have to do things multiple times for multiple research groups, that you can do it once and then in a de-identified way be able to spread your contributions out to many other groups. As we've been doing this, I think many of you know, and I'll go through the data today, you've been answering questions about what your child or your family member has been experiencing. I know sometimes I come back to the group to ask for refinements, because as I'm looking at the data that I'm seeing, I'm constantly asking myself whether this is teaching us something that we need to delve deeper in a certain dimension on. As we've been doing this, some of you know that you've had the opportunity, and we continue to encourage you to provide blood samples. Those blood samples are used then by researchers around the world to study CHAMP1. We can then be able to make cells out of that, and even, believe it or not, make cells that approximate or look like brain cells. And that's what researchers can use to be able to study CHAMP1 as they're trying to develop new treatments. It allows them to study those treatments in cells rather than having to test it out on people. And as I talked about with the gene therapy talk, it allows us to test to see whether or not the therapy is working, whether it's making the change we think it is, and if it's doing it in a very targeted way. So as we've been doing this, I think many of you know, we started out, believe it or not, we're at our 10th anniversary. So 10 years we've been at this. We've got many, many more years to come. And I want to give everyone the reassurance that we're going to be around for many, many, many decades, I'm sure, with this. We started out really as just literally sort of a few families together, and they've been growing over time. I know there are many, many families around the world, and this is an international community. So as, and I want to put my hat off to a few people, but Lindsay in particular, we have really emerged in terms of being able to support an international community that includes, obviously I'm speaking English, but being able to translate this into other languages so that other families down the road, and even if it's not in real time right now, but at a later time, can hear about this in many other languages. We're committed to helping catalyze researchers to be able to study your condition, CHAMP1, and to help guide them in terms of asking what are the important questions, what are some of the strategies that might work, but try and facilitate thinking about CHAMP1 in terms of its specific and unique features. So as we're doing this, I wanted to make some things clearer, because as I'll get to in a second, I know there are many families out there who haven't yet participated, and we want to make it as easy as possible. And if you have suggestions about where you've gotten stuck or where things have been challenging, please do let us know, because I'm sure we can make certain things better or easier, and definitely want to make this helpful to families. As we've been doing this, and you already heard about how to read your genetic test report, we do request as kind of your ticket to enter your genetic test report, because it gives us the specific genetic change, and I'll go over that in just a second. But it helps us to make sure you're actually in the right place. That is, that you belong in the CHAMP1 community, that's really you or your child's diagnosis, and that there's not something else going on. For the most part, people are in the right club, so I don't want anyone to get nervous that there's any major problem with your diagnosis. But I have to say that every once in a while, and that every once in a while is less than 5% of the time, but I will see someone that I either meet at a family meeting, or I'll start reviewing their records or their genetic test report, and I'll realize that they're actually not in the right group. And in some cases, we've been able to either reanalyze their genetic data or have them guide them to get additional genetic testing, and they've eventually found the right home, but we do want to make sure everyone gets to their right home. After individuals are admitted, so after we realize that CHAMP1 or confirm that CHAMP1 really is the right diagnosis, many of you have been kind enough to talk with our genetic counselors by telephone or video conference. I think most of the families find that useful to talk with someone who actually knows something about the condition. There are 2 genetic counselors who work with me, and one of them, again, we've made, she actually speaks several different languages, and so we've put Catherine Kentros, I just want to put out a shout out to her, has done a great job about trying to unite our families from around the world. She doesn't speak every single language, and so we do also have phone translators available for us if you happen to speak one of the other languages we're not fluent in. As we're doing that, we try and get a comprehensive medical history, and I'll be going over the medical history today. but we also have several other instruments. And these go into publications by researchers and help us compare CHAMP1 to some other neurological conditions and are going to be very helpful for us in the future as we're doing plans for clinical trials. So I talked a little bit about clinical trials in the last segment, the last video, and whether it's gene therapy or other things that we might use, we need to have a baseline to know what things are usually like so that we have a comparison. If we make a change, if we have an intervention, did we make a difference? Did we make things better? Did we make things worse? Maybe they're just the same, but we need to be able to know what that baseline is or what that, we call it, natural history is. So as we're doing that, we try and use as much as possible standardized measures, ways that are tried and true, ways of being able to, for instance, assess the brain, behavior, in certain cases, history of epilepsy. But we like to use things that are reliable, that other investigators will believe, have confidence in, and especially that have been used throughout other studies so that we have comparisons. So that's what we've put together, and a shout out to Leanne Green-Schneider. She's a child psychologist who's put this battery together with us, and she reviews all of the data. We clean it, we make sure it's actually that there are no mistakes or misunderstandings of the questions, so that as we distribute it to researchers, it's of the highest quality. We also come back to people over time. So we also want to see, as I said, this sort of longitudinal look, or seeing over time, do things stay the same? Do they get better? Do they get worse? We obviously have to actually check that over time. So we do have these longitudinal once a year check-ins, again, with the genetic counselors. We find that those are helpful. Again, families sometimes have questions. It ends up being more of a conversation in some cases, but to be able to get that information. As we're doing this, I also want to make the point that you as families are really important, and that we've learned a lot from listening to you, that you obviously live with this 24/7, and that there are some things that you're observing that may be very unique to CHAMP1 that I didn't appreciate. And I have to say, one of the things I really love about in-person family meetings is just to be able to meet you and to meet your children, your family members, because there are some things that I really appreciate much better by seeing, especially a room full of children or a room full of individuals, where I can even compare and contrast in my own head, make observations, ask you guys questions. And it was great when we had the family meeting in Florida, for instance, to be able to do that directly. I'll bring up one of the points a little bit later than I know some of you I've been conversing about back and forth and bring you up to date. This is specifically as a preview about the issue of cancer. And the bottom line is, don't be nervous, but I'll get to that in a second. So I'm going to present now the data on the Simons Searchlight registry of what we have from CHAMP1. As we did this, we had 42 individuals who originally registered. That was just a pretty simple, filling in online your email address. Of those, and I call this the funnel chart. So we start out with a large number of people, and it kind of funnels down to the number of people that I'm going to present data on you today. And the point is that we went from a larger number of people to a much smaller number of people. So as I said, one of the questions that I actually have for you all is there a problem in terms of either misunderstanding or difficulty accessing or having Wi-Fi or not speaking English, or anything that is a barrier in terms of 42 coming down to 12? Because obviously we want to be able to learn as much information as we can and share that with other people. And I should emphasize, all of this is completely free to the CHAMP1 community. No one in the group, none of the family members obviously get charged for any of this. Any of what we provide as a service to the community is free of charge. As we're doing this, I do want to just emphasize that what I'm going to describe to you today, I think the data are good data, but they're somewhat limited because they're only based on 12 individuals. At least for what I'm going to explain in great detail. And those individuals are on the age range of 2 to 26. And so, as you can imagine, I'm sure there are folks with CHAMP1 out there that are more than 26 years old. We simply don't have them in our group, and or they may not realize that's their diagnosis. That is, that they may be 46 years old, but no one's bothered thinking about genetic testing. And so they may not be accessing the resources to get into the club, so to speak. We've been thinking about creative ways to reach out to those individuals, figure out where they are, and even if we needed to offer mechanisms of free testing you know, we've done things like that before, and we certainly have, just a shout out, we have another study called SPARK, S-P-A-R-K, for anyone who has a genetic, or anyone who has a diagnosis of autism, to be able to join that study. And through that study, you can get free genetic assessment that will identify if they have CHAMP1 or potentially one of another condition, genetic conditions that are associated with autism. But with this, as I said, the numbers are somewhat limited. And one of the important things for me personally is, how does this change over the life course? So might there be different risks at the age of 36, 46, 56 than there might be in people that are 6 years old or 16 years old? Then I do think in the long term, that's some of the questions that I know I've heard you all ask that we want to be able to have the answers to as your children are aging up. So let me go over some of the details. You heard about how to read your genetic test report. This is not broken down in the form of a genetic test report, but we've taken all of the different types of genetic results we've from the families who have given us their test reports to review. And I'm going to put them in the class of, these are all things that disrupt the gene in the following way. All of us have 2 copies of the CHAMP1 gene. So we have one that we get from our mom and one that we get from our father. In the case of all of the CHAMP1 variants that I'm telling you about, we believe it's as if they have one copy of the CHAMP1 gene that's working perfectly fine, but the other copy of the gene, it's as if it's not showing up for business. So for all effective purposes, we believe that although in the DNA we can see 2 copies of the gene, at the level of the protein, or what comes out of the gene, effectively there's only half as much of the CHAMP1. Only 1 copy instead of 2 of the CHAMP1 doing its job. The other one, it's like it was AWOL, it didn't show up for work that day. So these are fancy ways, as you've heard of, in terms of the designations and of how we specify these. Don't worry about the specific details. I think at this point it probably is not a big deal, but I will just explain that where there are parentheses, those individuals are not unique individuals, but there are 2 individuals in this case, or 5 individuals in this case, with the exact same genetic variant. It's not to say that those people are related, so it's not as if you guys are brothers or sisters or cousins or things like that, because we think most of these, or in fact, the vast majority of these arise de novo or new in the child, are not inherited from either the mother or the father. So in this case, this same exact genetic change has arisen independently multiple times. And it may be that there's something in particular about that region of the DNA that makes that happen. But I think at the same time, all of these are acting in a similar way. That is, that there's only 1 functional copy of the gene that's working. So let me dive down into the details of the 12 individuals that we have across the age range. I wanted to show you just pictorially how old the individuals are that I'm telling you about. So again, as I told you, the age range goes from 2 to 26. It's predominantly, though, individuals less the age of 15. So we do have, again, 1 individual who's in their 20s, but otherwise we're really talking about kiddos here. So again, that is a limitation of what I'm going to be telling you about. Overwhelmingly, the thing that we see consistently for everyone with CHAMP1 is what I say is above the shoulders. So I think this is really primarily a condition that's related to differences in the brain. It manifests because the brain has impact on many parts of the body. So as I'm going to get into it in a second, I think we see other manifestations, but I think it's primarily a brain condition. In different individuals, that can manifest in different ways. Consistently, we're seeing that everyone has delays in their development of some sort, and we use slightly different terminology for this. For younger children, we call this developmental delay. And for older children, where the development is really mostly finished, we call it intellectual disability. So that really has to do with just how well we're able to measure this and how certain we can be that there's an issue. But I would say for individuals at all age ranges, we realize that they're slower in terms of learning, they're slower in terms of acquiring skills, and ultimately they won't reach the same attainment, for instance, as a neurotypical brother or sister. However, That does not mean that they regress. That does not mean that they can't continue learning and can't continue making progress over time. But as you're all aware, it takes more repetition. It takes more one-on-one. It takes special education to be able to continue that attainment. And I appreciate it's not easy. Some of the things that we see in particular are language is really challenging. And so with that, I also want to plant the seed that I have observed. Many of the children understand much more than they can verbalize and express. So in other words, their receptive language, their understanding, I think is better than their expressive language, their ability to tell things or be able to speak. I want you to be careful though that your children, like I said, are understanding and listening and absorbing like a sponge, all sorts of things around them. So be careful what you talk about. I'm being facetious, but I do think they're really great at being able to pick up many things, even some things that are not spoken, some, for instance, other body language and other communication. With this, as I said, we do know of individuals, though, who end up being, even as they're older, have real challenges with expressive language to the point of being minimally verbal. And I do encourage you to think about other ways of expressive language and communication. So whether that's by signing, picture board, communication devices, but I do think they're understanding the wheels are turning and they have things they want to express. It's just incredibly challenging to be able to get literally the sounds to come out of your mouth with the right different use of the tongue and the teeth and the lips and be able to get the phonation or the sounds exactly right. And so that's part of the challenge in terms of actually speaking and verbalizing. The other thing that, other points I'm going to make is that when the brain is already challenged, we want a full, clear pathway for information to come into the brain clearly. So what I mean by that is I want the children to be able to see as well as possible, and I want them to hear as well as possible. Those two senses are major ways in which they're experiencing the world around them, and we want them to have the most accurate perception of the world as possible so that they can learn and so that they're not afraid of the world, so they're not anxious because they don't understand what's going on around them. So with this, you'll see that many children do have issues in terms of vision. They can be many different things, and the bottom line is my recommendation, if you haven't already seen a pediatric ophthalmologist, especially if possible, a pediatric ophthalmologist for children with special needs, now's the time to do it. For those of you who have seen a pediatric ophthalmologist, I'm going to let them who know your child and their exact circumstance decide the frequency and the exact treatment, but we do see a range of different conditions. And so let me tell you a little bit about the range. So sometimes we have individuals like me who need to have glasses because they're farsighted. They can't see things far away and they need glasses to be able to bring those into view. I realize, again, that can be hard to even determine how well your child is seeing and if they can see at a distance. And so, as I said, those become critical to use a child or a pediatric ophthalmologist who's much better at being able to determine that when your child can't read an eye chart and call out those letters or numbers. We can also see things in terms of a lazy eye, and eyes that are not going and looking straight forward, but may veer off to one side. Sometimes that can be treated with patching. Sometimes it requires surgery, but important again to be able to make sure the eyes are aligned so that they're seeing clearly and seeing in the appropriate depth. Sometimes there are also issues in terms of not just farsighted, but nearsighted. The other issues in terms of the shape of the eye that can lead to an astigmatism. Again, all of those things oftentimes treated, very well treated with either surgery, patching, or glasses. We also see issues in terms of things that look more directly related to the brain. We're seeing essentially 100% of children with low muscle tone. Many of your children are receiving physical therapy. That helps, especially in terms of truncal tone, helps to keep your children strong, helps to keep them able to do more things. And long term, I'll get into it in a second, but especially the truncal tone helps to prevent scoliosis. So that's an important thing. One of the other things we've seen in about half of the individuals is a smaller head size. So we use the term micro, meaning small, cephaly, meaning head, microcephaly. For some individuals, their head size started on the smaller side, and as they grew up, it didn't shrink. So their heads continued to grow. Their heads did not shrink, but they didn't necessarily grow at the same rate of other children. And so as time went on, the difference between their head size and other children's head size became more obvious. And so they had microcephaly that became more apparent as they got older. In addition, some individuals have had problems with being clumsy, problems with coordination, and different people have called it different things. But like I said, clumsy, coordination disorder, movement disorder is something that we see. And then I don't know that it's exactly the right diagnosis, but people have described cerebral palsy. And that's, again, I think mostly a problem with movement and in some cases muscle tightness. So hypertonia in those cases. Thankfully, it hasn't been very often, but we have seen seizures. In some cases, I just want to tell you what to look for in terms of a seizure. Sometimes, thankfully, rarely, but this has been a grand mal seizure. This is now with sort of tonic, clonic, or what we call major movement, so arms, legs, potentially head moving or thrashing. Those types of things are not easy to miss. On the other hand, what we call petit mal or absent seizures can be very easy to miss. It looks like a child who may be daydreaming. They're kind of staring off into space, zoning out a little bit, but you can't sort of snap your fingers and bring them back. Sometimes, though, it can be hard to tell. And so if you're not sure if your child is having a seizure, if you do have a smartphone where you can take a video, I think it's a great way of being able to capture the moment and then being able to share it with your neurologist or pediatrician, have them give you some independent assessment of whether or not they think it's a seizure. And then ultimately, if it's something that's concerning enough, we generally bring children into the hospital to have an electroencephalogram. So at that point, they put electrical wires onto the head. They can measure the electrical activity. And my recommendation is usually to do that in the hospital over a 24-hour period, including overnight, because there are some types of seizures that you won't see except at night. And for many of you, I expect you're sleeping while your kids are sleeping, so you're probably not sure what's going on while they're sleeping. But by doing that with the video EEG, if you're seeing something, one of the benefits is we have in the hospital a button that you can press, and then that synchronizes with the video and with the EEG. So if you see something that you're concerned about, it's a good way to answer your question. Was that a seizure or wasn't it a seizure? So it's a good way, as I said, to be able to try and capture what we can. Now, one of the problems is that even being in the hospital 24 hours, it's not like you're there a month, and if a seizure or whatever that is infrequent, you might not happen to catch it when you're in the hospital. But if something's fairly frequent, then we should be able to see that. Another common condition we hear about from families are tummy issues. So they're gastrointestinal issues. You may not have been thinking about it so much, but the brain also has nerves that go to the intestine. And in the same way that the brain's not functioning for other parts of the body, like the muscles or the eyes, in the same way it may not be functioning to the gut or to the intestine. So frequently what we'll see is that the intestine goes through a process of literally contracting and moving when you eat things, moving it through the intestinal tract so that it comes in the mouth and obviously it goes out the other end. If the contractions are slow, or if there's essentially hypotonia of the intestine so that things are not moving along so fast, you can end up with constipation. And in fact, that's what we see with many of the children. On the other hand, if you have that hypotonia or that low muscle tone, instead of things sort of being a one-way valve after you eat and have it going into the tummy, that one-way valve may be loose because that muscle tone is loose and things may go back up the other way. And then you end up with a problem that we call reflux or heartburn. And so both of those we see quite frequently. We'll even have some children that will alternate between diarrhea and constipation. Sometimes things just seem, and sometimes it's certain foods, just to seem to flow right through. There's no real magic treatment for this, although I will say it is something that often can be treated either by diet, certain changes in the diet, And honestly, that's a little bit of a trial and error. I do urge you to keep a diet diary so that you can, not just based on your memory or your recall, but actually write it down and see if you see any pattern with certain types of food being associated with certain types of problems. And then a pediatric gastroenterologist can often be quite helpful with medications that help things move through the system faster, stool softeners, colace, or when we need to, enemas to be able to clear out the system. But again, if your child is uncomfortable, it'll often affect everything from learning to just, mood and how they do all through the day. I put through a list of the surgeries that children had, mainly to reassure you that for the most part, they have not been serious problems. And so the reason I bring this up is surgery to me is kind of a big thing. It has to be a serious enough condition to get surgery, but most of the surgeries I have listed here to me are relatively minor and common pediatric surgeries. So getting ear tubes or getting your tonsils out or having your eyes straightened, as I talked about, most of these are pretty simple. Having a hernia repair, I've had that, so pretty standard. There are a couple here that I'll point out though. One is that there was, you may have seen it on the other slide, there was one child who had a malrotation. That's more of a congenital or a problem present at birth, and that required a more sort of intensive surgery. That's not very common. I want to point that out, just one child. And then I'll get to in a second, but one child had a bone marrow transplant as the treatment for their leukemia or their cancer. So with this, let me take 1 second because this was something that I know I had asked the community about. So let me explain why this happened. So I've known for a long time that we've had one child in the community with leukemia. And I've been, that was one child, and I wasn't sure, was that just one child? And, it could be 1 out of 1,000,000 or it could be 1 out of 10. And just when it comes to small numbers, sometimes it's hard to tell at the beginning if it's going to be a condition that we're going to see quite frequently or quite infrequently. So I became more concerned when I heard about a second child with cancer. And second child, given, as you've been seeing, our numbers in the community are pretty small. One could be just 1 in 1,000,000, but if we saw 2 when we were only seeing about, less than 20 kids in the community, that started getting me more nervous. The bottom line is that for the second case, I'm not yet sure whether or not that particular person belongs in our CHAMP1 club. So I talked about this at the very beginning, how we're so careful to make sure that anyone who's in the club really should be in the club, because we want to make sure we're giving you accurate information. For that second case, and I'm not calling anyone out, so I hope no one's uncomfortable with this, but looking at this a little bit more carefully, I think there's still more work that we need to do to determine whether or not that person belongs in the CHAMP1 club. And then based on that, I'll be able to determine much better whether or not cancer is a condition that's associated. So why am I so worried or paying so much attention to the cancer? Number one, of course, it's a serious condition. And if we do need to be screening by looking at a blood smear every year from your children, that's important to know. That's an easy enough thing that your pediatricians could be doing if that's a real concern. The other part about this is this particular gene, the way it works is by lining up the chromosomes. So the chromosomes are the sticks that contain the genetic information, and this gene works to line up the chromosomes and that makes sure when the chromosomes divide, they do so correctly. Now, one of the things that happens with cancer in some cases is there can be disruption of the way those chromosomes divide. And so one thing I've been worried about is because of the fundamental way that the CHAMP1 gene works, could it disrupt these chromosome alignments and separation in a way that might predispose then to leukemia or to other cancers? The bottom line in this is I don't have a final answer for you. It's something that I still very much would like to resolve as best we can, because I do think it's one of the things that has important medical implications for the community, but I don't yet have an answer. And if any of you are hearing about anything, certainly let me know. Recurrent pneumonia is something that's been a little bit more frequent, but I'd say it's not something that I'm especially worried about, again, a little bit more frequent within the era of SARS-CoV-2 or COVID-19. We're paying especially a lot of importance to children who are getting infected in general, but specifically with SARS-CoV-2. I haven't heard of enough infections in a good way. I haven't heard enough infections in the CHAMP1 community to think this causes specific problems, but it is something, some of you may have gotten a survey, in fact, from Simons Searchlight. It's something we've been asking the community about, and thankfully I can be mostly reassuring. We have seen a few issues here and there, so kind of onesies and twosies, issues with people being on the shorter side, girls with the irregular periods, some issues with weight gain, but they haven't been frequent. And again, remember that age distribution I told you about. We are starting to see, as children are aging up into teenage years or getting sort of tween years, perhaps some scoliosis or curvature of the spine. We frequently see that with neurological conditions, not only CHAMP1, but more broadly with neurodegenerative conditions, especially as children go through rapid growth and the spine not having as much central support. So physical therapy helps with this, and screening helps with this. So making sure that your pediatrician is checking for scoliosis, something easy to do in the doctor's office. Within this, another measure of severity is whether or not people needed medication to be able to treat the condition. So again, we've put together the list of medications or conditions for which people use medication so that you have some sense of how frequent, how serious. As I said, if we do see epilepsy, we do tend to treat it medically. And so I think that's what you're seeing here. And for some children that were having behavioral issues, that's something else that comes up. I'm guessing it also may change over the life course. That is, we may see a different set of behavioral issues for younger children than for adolescents, than potentially for adults. So again, it's something that we'd like to be able to understand how this changes, and we're looking at our longitudinal data. With this, we always take a sort of deep dive in terms of medications for epilepsy or potentially other things besides medications that people use, because that's one that has significant medical impact. And we don't want to over-medicate so that a child becomes sedated or sleeping, and we don't want to under-medicate such that they don't get the medication to treat the seizures. The bottom line is that there's no clear winner in terms of like one medication that was the absolute hands down favorite, but we have listed, and again, all of these slides are available after the meeting, listed the medications that children have taken. But again, there's just such small numbers, I think it's too early to say anything definitive. So in brief, and I'll wrap up here to say that mostly what we're seeing are things that are related to the brain, but things that are related to the brain, behavior, development, eye issues, muscle tone issues, and then also some gastrointestinal issues. We do see autism, although it's not 100%, certainly it's a minority of individuals with autism, but we can see some of the behavioral features that are autistic-like symptoms in more of the children. So finally, if there are any researchers watching this today, I just want to highlight, this is the URL, safaribase.org, or rather I should say base.safari.org. This is our Safari Base portal. For researchers, it's very easy to make an account and put in a query and fill out a research distribution agreement to be able to get the de-identified data. And for those of you who might need induced pluripotential stem cells, you're also able to order that through this portal. So I will stop here and we can answer any of the questions that might have come up through.
Okay, so thank you so much, Wendy, for that presentation. The first question that we had was actually during the lab report presentation, and this person is just saying they're one of the first families to receive a diagnosis in 2015. Do you think it would be worth asking for another test? We only received a one-page report.
Okay, so for that one, it may be one of these questions we can take offline, and we're glad to do that. For individuals who had their testing by exome sequencing, and from something like 5 years ago, that might have been the case. Exome sequencing is a very comprehensive test, and if you were identified to have a de novo or a new genetic change, so not inherited from mom or dad, but a new genetic change in the child, and especially if it looks like it's one of these types of variants I put up on that slide, where it's got one of those little stars or an asterisk, that's the easiest way to see it, or the letters FS for frame shift. If you fall into one of those cases, then this is probably the answer and no other testing is necessary. If you did have exome sequencing and you're worried or you're wondering if there's something that's changed, you can also ask your doctor to ask to have the test or the information reanalyzed. So you don't have to do a new test over from start to finish. They can just analyze the data one more time just to make sure there's nothing new or different. So I hope that answered the question.
Thank you. And the next question, what does gene therapy consist of?
Okay, good question. So gene therapy can consist of many different things. I think the question you're probably asking, but you can certainly clarify, is from your point of view. So if you're the one getting gene therapy, what happens to you? What happens to your body? So there's not one answer because it depends on exactly what gene therapy we're using and where we're trying to go. I'll give one example, and I'm saying for the CHAMP1 community, I don't know how this is going to play out. For SMA, I mentioned that in my talk. For spinal muscular atrophy, the way that works is it's an IV infusion. So it happens over a couple hours where we just put an IV in and do the infusion of the gene therapy. And from there, the gene therapy goes to specific types of neurons or brain cells that we needed to go to, but that's it. It's like literally a one and done treatment. It happens over a couple hours. We watch people carefully before and after, but that's all that's involved. There's some other types of gene therapy I alluded to a little bit. In those cases, we actually take cells out of the body. We treat the cells outside of the body with the gene therapy, and then we give those cells back to the person. So they're their own cells, but we change them outside of the body. And that's another form of gene therapy. That's a little bit more involved. And with that type of gene therapy, which I'm not saying is what would happen with CHAMP1, so I'm not going to describe it in too much detail, but there's more in terms of hospitalization that goes on with that and for a prolonged period of time. There's a third type of gene therapy that we're doing with other neurological conditions where, as you can imagine, we have to get it into the brain, into the brain cells. And to be able to get it in there sort of with enough power, getting it into enough cells and making sure it's really effective, we actually do direct injections into basically the middle of the brain. So there's a spot we can do in the brain called the ventricle that has some of the fluid that bathes the brain. So we do injections through a little hole in the skull directly into that ventricle and allow it to then, that virus to be able to get into all the different parts. And then one other way that we do this is to be able to get in the same way, but instead of doing it through the skull, we do it actually as an injection into the back. So right around the spinal cord, and then it can float up through that same space up into the brain. So a few different things. The bottom line is that I'm not saying it's going to be any one of these specifically. I don't know yet is the answer in terms of whether this is just going to be an IV, whether it's going to be an injection to some other part of the body. But certainly we're going to try and do what is least painful, less invasive and safest, but also most effective. So stay tuned.
The next question asks, how would gene therapy help with dystonia all over all functions?
Okay, so in general, the thinking, at least with gene therapy, is that this would replace that, again, I talked about one copy of the gene not showing up for work. If gene therapy is going to work for CHAMP1, probably what we're going to do is try and deliver back a copy of the gene that's working so that we'll get the full amount of CHAMP1. With that, we hope to normalize the way those brain cells are working. So if dystonia is because certain brain cells are firing or constantly active and constantly working overtime because of the missing CHAMP1, if we replace that CHAMP1, can we allow them to take a coffee break? Can we allow them to restore normal function, be able to fire in the normal way, be able to connect and send signals the normal way? So that's the hope, is that we'll be able to correct those deficiencies. There's still an open question, as I was alluding to, in terms of if you, say you do gene therapy when someone is 10 years old, will you suddenly make them a normal 10-year-old? Probably not. It's not like overnight you're just going to fix everything. And I think it's still to be determined how much of that we'll be able to repair from the, the preceding 10 years, for instance, how much we can play catch up and how much we can be able to normalize function. So I don't know the answer to those yet. Certainly we're trying to move as quickly as we can to give the option to the most people as we can at the earliest age we can. But these are a lot of open questions.
The next question, is there any specific diet we should be following that you feel will help with CHAMP1 to keep the improvement up? Or is there anything special we need to do to keep the function up once the medicine is given?
Yep. So I'm going to answer this in a relatively generic way because I'm not sure exactly which medicine and other things. So a couple recommendations, and I'm saying this specifically because of the time and history where we are right now. So for CHAMP1 folks, I think they benefit a lot from being able to work with their therapist, whether these are physical therapists, speech therapists, special educators. They derive, I think, a lot of benefit from that, both in terms of physically staying strong, keeping good muscle tone, being able to help with the speech and communication. But I realize under COVID situations, at least for some period, undoubtedly you were doing things at a distance or they were just shut down completely. I know it's been a lot in the news, especially even within the last 24 hours in terms of now this resurgence of COVID. And it's definitely the case that as we've gone back to school in the United States, at least, there are certain places where we are seeing an uptick within the infection, the virus in communities. And in some places, people have just been on distance learning the whole time anyway. The reason I bring this up is because I do know that at least my experience with my own patients is some of the children have either plateaued or even, I think, lost some of what they had before. I don't think it's an inherent problem with CHAMP1. I think it's just that we haven't been there to reinforce and continue the therapies that we started, and practice makes perfect, and the kiddos just haven't had as much practice. I would encourage you, this is always a balance of risk and benefit. And so not knowing each of your own individual situations, I'd say talk to your doctors and therapists about how to balance that risk and benefit. But it is important for the kids to be able to get some type of real therapy. And that oftentimes requires being in person, even, for instance, a speech therapist to see how the lips and the mouth and the teeth are moving and help to be able to shape and form and touch. But that, obviously is associated with issues with masks. You can't see how lips are moving if you have a mask on. You can't help in terms of many of those personal lessons. So there are ways through frequent testing, through frequent monitoring of symptoms, through being able to figure out what the risk level is in your home, being able to do things that are one-on-one and, minimize the number of contacts, and of course, use lots of hand sanitizer. There are ways of being able to do things in a COVID safe way, or at least COVID safer way. And also I apologize because I know, especially for the parents out there, you're doing double duty. Many of you are working your regular day job and you now have become, teacher mom, teacher dad, or therapist mom, therapist dad. I know it's extra draining, but it is important to try and take as many tips as you can from the therapist and try and reinforce that evenings, weekends, days that they don't get in-person therapy as much as you can for that. So that as you're doing this, as I said, I think that probably is going to make a bigger difference than anything right now specifically. Otherwise, in terms of diet, I don't know of any magical diet where, if you just put in, for instance, fish, if fish is your main staple, that all of a sudden all those fish oils are going to make all this better. I don't know of anything like that, but I do know that many of the kiddos do have problems with more or less constipation with certain foods. So whether it's dairy, whether it's things that have gluten in them, various things can be a trigger for kiddos in terms of GI problems. And those GI problems do oftentimes tend to spill over into other behavioral problems with discomfort that they can't communicate. So like I said, keep a diet diary, try and keep a log. And I'm sorry, I don't have a quick fix for this. But like I said, some of those triggers can be dairy or gluten.
Thank you. And this next participant says, my daughter and I are both heterozygous for the CHAMP1 gene, but we also have variant of uncertain significance. So I was just wondering, is there a higher chance with passing it down since I passed it down to my daughter?
Yep. So this is something that one of our genetic counselors is happy to go over with you, look a copy of your test report and be able to help you, as I said, with that verification that CHAMP1 really is the diagnosis. And we'd be glad to do that with you offline one by one or on a one-on-one basis. A variant, let me just explain the nomenclature a little bit. I know you heard this on the earlier recording, but a variant of uncertain significance means that we don't know. Most variants of uncertain significance end up not being the answer in genetics, so don't jump to the conclusion that a variant of uncertain significance equals a diagnosis. So that's the first thing. In general, for situations like this family, if a parent has this CHAMP1 change, so I'm just going to call this change, this is the way my fist is balled up. If a parent has this change, then it's a 50-50 chance of whether or not they pass on the normal CHAMP1 gene or the one with the change. So it's flipping a coin 50-50. That's true whether you have a daughter or a son. It doesn't really pay attention to sex in that way, and it should be 50-50 all the time. On the other hand, the reason I'm putting sort of a note to self on this is that most families, this is not something that gets passed down from a parent to a child. As I was saying earlier, for the majority of people, this is something that starts out brand new in the child. We can see it pass down, so I'm not saying that this is wrong, but again, it's something that we look at extra carefully just to make sure this really is the right diagnosis.
Thanks. Next question. Are our children more susceptible to low insulin production or problems with hypoglycemia, or hyperglycemia?
Okay, so that's a good question. To my knowledge, I have not, with my own personal patients or families who've reached out to me, seen evidence of repeated either hypoglycemia, so low blood sugar, like where someone would pass out or have a seizure or lose consciousness, or hyperglycemia. So hyperglycemia is basically diabetes. So I treat children that have other problems with insulin production, other sort of diabetes related things, but we've never seen this come up in our medical history interview in terms of any of the data that we've seen. Again, we've got limited numbers, so there could be something that you're seeing that just hasn't been reported to us yet, and do feel free to register and let us know if that is something, especially if more than one family is seeing the same thing. But no, I haven't seen that as being a consistent feature in CHAMP1.
Okay, and the next question, are our children's digestive and constipation problems caused by low muscle tone in the gut, or is there some other cause?
Yep. So I can't say that this has been scientifically definitively proven. So it's not like we've taken, sort of any people and fixed the problem in one part of the brain and not the gut part of the brain, or, done any fancy science fiction-y things like that. So I can't say for sure, but I will say that it is very, very consistent across neurogenetic conditions, not just CHAMP1, to have these same symptoms that I talked about. So in general, we think it is something that's related to essentially the brain in the gut, the nerves that actually innervate or come down from the brain to the gut. And it's a very complicated system. I'm glad to talk with anyone about it if they're really interested. At the end of the day, though, I do think if we could do something to fix the brain or fix this centrally, it would significantly improve the problem. And in the meantime, as I said, things that we can do in terms of some of the relatively benign medications, common medications to use, as well as some of the dietary changes. There are also some other tricks in terms of keeping your kids active and some, things about patterning of toileting and things like that that can help as well. But those are not specific to CHAMP1. So if you're getting advice from your pediatrician about other things, they're probably generally true for CHAMP1 as well.
Thanks. Does having a different blood type from the mom and dad play a part in CHAMP1?
So the short answer to that is no. So the blood type, whether it's ABO or whether it's positive or negative, completely different genes, different places within our genome. So that one's unrelated to CHAMP1.
Great. And does CHAMP1 cause slow brain waves?
So that's an interesting question. We formally have not looked at the brain waves for individuals with CHAMP1. So there are ways of, as I said, you can look at the brain waves directly through an electroencephalogram. There are different, it's a very complex way. You can look for spikes and you can look for other things that we see in brain waves. It doesn't necessarily correlate that slow brain waves equal slow learning. So I just want to be clear about that. You can have other problems with the brain waves that also cause slowness in learning. And I think probably at the level your question is asking, it's a great question. If families are willing to donate their EEGs or their electroencephalograms, if this is a question the community has, we'll be glad to either, collect the data and make sure that scientists can start answering that question, or, if we get enough of them, it's possible that we could, specifically ask a scientist to look at that. But I do not know definitively the answer technically to what the EEG looks like for all of CHAMP1.
Okay, and this question or comment came in two parts, so I will try to do it justice, but I think they're saying there's a third argument for leukemia risk. There's a Japanese study on precancerous blood cells seen in a CHAMP1 mutated subject.
Yeah. So this is part of the thinking, again, for those of you who I'd sent this message out in terms of looking for anyone else in the community with leukemia or anything else. It is all of this data together, as you said, that has made me, I'm trying to, not make people nervous or anxious when we don't need to be, but I'm also trying to explore to see if we can understand this better. So it is still an open question for me. Again, I want to be clear that in terms of when I had the call out to see if any other families had seen leukemia or cancer, I don't definitively have a second case of CHAMP1 with a full-blown cancer yet, to my knowledge. I'm not saying we're off the hook, but I'm just saying that's where we are at this progress right now.
Thank you. The next question is drug repurposing a better option than gene therapy for our kids?
Yep. So the idea with drug repurposing is that you may be able to use a medication that's already, for instance, been approved by the FDA for another indication. This is an appealing strategy because if we have a large amount of experience with it from a safety point of view, we might feel really safe giving it to our kiddos. So, I'll just make this up, but, if you had something that people had used for high cholesterol and millions of people had been exposed to it and didn't have problems, we might all feel very comfortable if we had some evidence it was going to work for our kiddos with CHAMP1 to be able to give that medication to them. That also means you don't have to go through long years, large numbers of years trying to demonstrate it, manufacture it at large scale. People can quote unquote prescribe it off label in those cases. So yes, there are researchers. I don't know of anyone in the CHAMP1 community specifically, but there are researchers and there are what we call high throughput drug screens to be able to do such studies and to screen and check and see if there might be medications that could be tried. These are done commonly for FDA approved medications or for other medications that maybe didn't get to the point of being federally approved by the Federal Drug Administration, but that different pharmaceutical companies have within their libraries. The only limitation is that, like I said, for this one particular gene that we're talking about, CHAMP1, there might be another medication that might be able to increase how much is produced, but I'm not expecting that a medication alone would be able to completely get this back to normal. So it might make things better, and that certainly would be a very good thing, and no reason not to use that if we can. But I don't know that it's going to be a cure with a capital C. In other words, completely ameliorate this with simply using a drug that's already been made for another purpose.
Thanks. Would it be safe or beneficial to supplement for gut health with tributylene and nucleotides?
So it might be. So, some of the things that people have tried are, I'll also give some other alternatives, probiotics. So some people have used, we have bacteria that are in our gut. And so some people have used probiotics, especially I'll say after someone's been treated for an ear infection. And the reason I mention this is with an ear infection, you take antibiotics. The antibiotics can change your microbiome or the gut or the microbes in your gut. And with that, trying to get back to what they were like before is something that sometimes helps. With this though, I don't know of anything that's like in terms of a supplement that's been, great for everyone in terms of this. I don't specifically have evidence of what you had mentioned, the tributylene, at least in people in the Simons Searchlight study having tried that, or any evidence in terms of whether it worked or not. So I don't have any specific data to say, yeah, it's great or no, it's terrible. But like I said, what I see most commonly is people that are doing dairy, gluten in their diet, or as I said, some of the commonly used medications either for reflux or constipation.
Thanks. This next question is concerning gene therapy. We don't know for sure if it's a problem of haploinsufficiency or abnormal protein produced due to the CHAMP1 mutation, do we?
So that's a very sophisticated question. So if you go back, I'm not going to do it right now, but if you go back to the slide that I had, all of the different mutations that we've seen that we think are problematic are all what we would call disrupting changes. So they all seem to lead to a truncated protein, but they tend not to do it in any one spot. That is, that we see truncations really throughout the protein at different locations. And in general, when I see a mutation distribution that looks like that, it's almost always the case that those are due to haploinsufficiency, or what I had talked about. That's a fancy scientific way of saying only one copy of the gene is working and the other copy of the gene doesn't show up to work. So while that hasn't been definitively proven 100%, dollars to donuts, I'll bet you $1 on this one, that I'll be right that this really is a haploinsufficiency mechanism.
Thank you. And this one, just, are people with CHAMP1 at greater risk during COVID-19?
So we don't technically know the answer to that. What I've observed though, like I said, from the survey that we put out to our Simons Searchlight community and what I've been seeing as a pediatrician generally, is that the answer so far is I don't see any risk that they are, or I don't see any evidence that they're at increased risk. Here's what I tend to see in general. Our kiddos, oftentimes any illness, so even if it could be the cold or the flu or any other viruses, they tend to be slower to get better. That is that it takes longer to recover, and they have more problems in terms of just sort of picking up where they left off after the infection. So I do think the infections can be a little bit harder on them in general. And this is true of all children that we know about so far. And I can say at Columbia, I think we saw more children and had more experience than almost any place else in the world. Children are quite resilient to the infection. Even those that get this post-inflammatory or this systemic inflammatory condition afterwards, we're very, very well able to treat them with steroids and something called intravenous immunoglobulins. So the children recover. We haven't had any deaths at Columbia with that treatment that we've used. And so even if they should get sick or even have the post-infection complications, they've been doing fine after that. So again, I don't have any evidence that SARS-CoV-2 or COVID-19 has been especially dangerous or problematic or problems in terms of recovery with CHAMP1. Where we have seen issues, which again is not the CHAMP community, has been in individuals who have pre-existing lung conditions especially, or some individuals with pre-existing heart conditions, but simply neurodevelopmental conditions hasn't led that I've seen to an increased risk of stroke in the children, for instance, or any of the other SARS-CoV-2 complications.
Do we know if a lot of the characteristics or symptoms of CHAMP1 are predominantly present at birth or early age, or continue to develop and progress as the child ages?
Good question. For many of the symptoms, they may not be present at birth. For those of you who are moms or dads, I often will hear you say to me that I knew something was different early on, whether it was exactly the day they were born or whether it was in the first few months of life. But you often tell me when I talk to you that you just knew something was different. Either the muscle tone was lower or how they engaged with you. So we may not have been able to see everything at birth because like children don't come out the chute talking, right? So it's, we can't see deficits in talking at birth. They only become evident a little bit older as the children miss the milestones that other children are doing. The same is true also of the microcephaly, or the head size for some of them. So as I was describing, for most of them, their head size is a little bit smaller than other babies. And then what becomes more obvious is over time, there's a greater discrepancy between their head size and the other children who are the same age. So I think we tend to see that. Again, I want to emphasize this because it's an important point. As far as we know, this is not a degenerative condition. This is not a regressive condition. This is not something that gets worse over time. The limitation to everything I've said, though, is, like I said, our oldest people are still young adults in the community. So how this looks like, what it looks like in the 40s, 50s, that I think is still an open question.
Thanks, Wendy. And we're almost out of time, but we've just got 2 questions, so hopefully we can hit them both. The first, do you think it's necessary to have an MRI of the brain or an EEG if there is no current concern?
So the MRI, no. It oftentimes requires sedation and it's not necessary. The EEG, electroencephalogram, I would say if you haven't had one at baseline, it's a good idea to do one. Especially if you're seeing anything that looks like those movements. I explained that I was concerned about the staring off in space or any type of jerking movement.
Thanks. And the last question, do we know exactly what the CHAMP1 gene does?
Yeah. So the CHAMP1 gene, I'm sorry for the technobabble, is a kinetochore microtubule attachment protein. So it does what I talked about before. That is that your chromosomes need to line up and they need to pair, and then they need to separate, and they need to separate evenly when cells divide. And so this is a protein that's responsible for that process of those chromosomes coming together, lining up, and then dividing as cells go through the cell division. With that, we sometimes see with other genetic causes of microcephaly, we have seen other proteins that are in that same family of CHAMP1 that is responsible for the kinetochore and those chromosomes lining up and dividing. And so this makes sense to us from a biological point of view. And as I said, sometimes that chromosome lining up and division, when that's aberrant, it also can be related to leukemias or to cancers. And that's why we've been especially, looking at that carefully and trying to monitor that situation so closely.